Welcome to another series of Study With Me But I Teach You video. Today we're gonna delve into the amazing world of biochemistry, and we are especially in the chapter 7 of Lehninger's biochemistry book, Carbohydrates. Let's dive in together. Hello everyone, as you know, I am Inci, a third year molecular biology and genetics student at Acıbadem University, and I started this series to go over the concepts that I might come across in my exams, and today we are doing that. This is one of my midterm topics on biochemistry, so let us learn together. When you look at a pure sugar, what do you see? A colorless crystalline solid, and when you drop them in water, they dissolve. But what if you drop them in a nonpolar solvent like ether? Nothing happens. Why? Because of their backbone. The simplest sugars are unbranched carbon chains covered in hydroxyl bonds. These hydroxyl bonds are loving to make hydrogen bonds with water. That makes it dissolvable in polar solvents. But what gives a sugar its ultimate identity? The carbonyl group. If the carbonyl group is very head of the chain, it's called an aldose. If it's hidden inside the branches, like this, it's called a ketose. Now hold up your hands. They are identical. They are mirror image of each other, but you can't superimpose them, and it's called chirality. Molecules do this too. If we look at the chiral carbon, the furthest from the carbonyl group, if the OH points to right, it's called a D-isomer. If it's pointing to the left, it's called an L-isomer. Here's a biological mystery. Almost every naturally occurring sugar in the universe is D-isomer. And all enzymes, like a molecular glass, are made to fit them perfectly for these right-handed sugars. And what if these sugars differ in only one chiral center? All the other structures are the same. They are just differing in one chiral center. We call them epimers. D-glucose and D-mannose are epimers of each other at the carbon number 2. And when we look at D-glucose and D-galactose, they are epimers at the carbon number 4. And one tiny little flip changes its entire biological function. But here is the golden rule of biochemistry. If a sugar has more than 5 or more carbons, they never generally stay in their linear form in your cells. It does something like a molecular yoga. The oxygen atom on 5th carbon attacks the carbonyl group, and when an alcohol attacks an aldehyde, it forms something called hemiacetal. If it's ketone, it's called hemiacetal, or it's called hemiacetal for ketosis. And glucose forms a 6-membered ring. It resembles a molecule called pyrene. That's why we call it glucopyranose. And fructose forms a 5-membered ring resembling the molecule called furan. So we call it fructofuranose. When this ring shuts down, the first carbon is called as anomeric carbon. If its new OH group points down, it's called alpha. If it's pointing up, the OH group, it's called beta. In our blood, These glucose chains is constantly opening up and closing down, flickering between alpha and beta. We call this Muto rotation. But wait, textbooks draw this as a 2D structure of an hexagon. That's a lie. The carbon atoms actually pucker up and down to relieve the steric strain, following a 3D structure that looks like a long chair. This chair conformation is the true geometry of our sugar molecules and our sugar molecules' sweetness. And nature loves to build. When two sugars connect, they release a water molecule through a reaction called condensation to form an or glycosidic bond. A laboratory secret. This specific bond is easily broken by a boiling acid, but it is completely immune to base. Let's look at maltose. The first sugar is locked, but the second sugar, the anomeric carbon, is totally free because it can still open up. It can donate electrons to oxidizing agents like copper ions. If you put cupric ions in a test tube with maltose, the sugar reduces the copper to cupreous ions. We call this a reducing sugar, and this is how we historically tested diabetes for urine. But when we look at the table sugar sucrose, the bond is alpha-beta, meaning a glucose and a fructose is coming together to make up sucrose. And both anomeric carbons are locked in the bridge. This is called a non-reducing sugar. Sucrose and the insect block sugar, trehalose, are non-reducing sugars. They are like a molecular safe. Let's scale up polysaccharides. Plants store energy as starch, specifically amylopectin. It's a long alpha chain that branches every 24 to 30 residues using a 1 alpha to 6 carbon bond. But animals, we store energy as glycogen, and surprising, glycogen is exactly the same, but it branches every 7 to 12 residues. But why so many branches? Because of evolution. More branches mean more non-reducing ends. When you need to sprint away from danger, your enzymes have millions of ends to cut these glucoses from. It's like an explosive energy release. But if you want structure, you need to switch to beta configuration. Cellulose is a linear chain made up the glucoses linked by beta first carbon to fourth carbon. Because of this beta geometry, the chain doesn't coil. It stays perfectly straight, forming unbreakable hydrogen bonded networks. Like, think about it's wood. You can't use an enzyme to easily break it. And what about insects? They use chitin. It's like a cellulose, but we replace carbon to hydroxyl group with an N-acetyl amino group. Bam! You have a hard exoskeleton of a spider. When it comes to bacteria, they use peptidoglycan, a heteropolysaccharide. It's made of alternating GLC, NAC, and MUR2AC sugars, cross-linked by tough little peptides. It's like a molecular bulletproof vest. From the simple mirror image of a carbon to the flickering mutation of the pyronese ring, to bulletproof vests of bacteria and structural pillars of giant redwood trees. Carbohydrates aren't just what you eat. They are most versatile, complex, and most beautiful building blocks of the universe. And this will be the end of part 1. See you in the part 2. Welcome back to the part 2 of our masterclass in biochemistry. In part 1, we built the carbohydrates. And for this part, we are going to dive in their secret language. When we look at every cell in our body, it's not a completely smooth sphere. It's covered in dense, forest-like microscopic sugars called glycolysis. When a sugar covalently binds to a peptide or a lipid, it forms something called Glycoconjugates. And this is how cells communicate. Let's look at the 3 major players. Proteoglycans. These are the massive macromolecules in your extracellular matrix. They are mostly sugar, glycosaminoglycans. These sugars are attached to a core protein. But how do they attach? They don't just stick randomly. They use a highly specific tetrasaccharide bridge, glucuronic acid, and that X-lows binds to the styrene of the protein. This is molecular engineering. Secondly, glycoproteins. These are mostly proteins. Think of your antibodies. The sugars can be all linked to serine or threonine, or could be unlinked to amine nitrogen of asparagine. And glycolipids. These are lipids with sugar heads. Think of a lipid bilayer or LPS. If we look at the gram-negative bacteria of Salmonella, its outer membrane is covered in lipopolysaccharides, LPS. It has a lipid A tail, a core oligosaccharide, and all specific chains. This exact molecule triggers a massive immune response when you get a bacterial infection. But who is reading these sugar codes? The answer is proteins called lectins. Lectins are biological barcode scanners. They bind to specific carbohydrates with extreme precision. Let me show you a real-life molecular miracle. Leukocyte rolling. Imagine you get a cut on your finger. Bacteria enter and your white blood cells, neutrophils, are rushing to that cut through your bloodstream at high speeds. And the question, how do they know where to stop? The endothelial cells near the cut put a stop sign near that cut. That stop sign is a lectin called P-selectin. As the white blood cells flies by, P-selectin grabs them from their sugar ligands, but it's not like a permanent grip. It just grabs, then releases, and causes the white blood cell to literally roll along the wall, slowing down. And finally, a stronger protein called integrin locks it down, adhesion, and the cell squeezes through the wall, extravasation, to find and fight the bacteria. This is all orchestrated by the sugar code. A fun fact, toxins use this too. The cholera toxin specifically binds to GM1 ganglioside sugar in your gut. The influenza virus flu uses hemagglutinin to bind the sialic acid in your cells. The sugar code can save you or can kill you. Works in either way. Lesson. This was the end of our carbohydrate study session. In the next part, we'll enter the world of Glycolysis. Till then, please don't forget to subscribe to my channel. Check out my other videos. They are mainly in Turkish, but I'm improving myself on speaking English and I'll be better, I promise. I get more confident each time through each video. It will all work out for the better. And everything that I mentioned in this video will be in the description box down below. See you in the next video. Thank you for watching this one and studying with me.